So these excitatory postsynaptic potentials, or EPSPs, right? And these inhibitory postsynaptic potentials, or IPSPs, are local potentials. They're occurring at individual synaptic connections, individual axodendritic synapses, and also axosomatic synapses, and occasionally at axoaxonic synapses. And an action potential, in contrast, is something that is initiated by reaching threshold at minus 55 at the axon hillock, and then opening all those voltage gated channels down here, that can really set off an all or none kind of propagation of an action potential from the hillock all the way down to the end of the axon to the axon terminals. So you have the cell. It's got an interesting job. It's a remarkable piece of machinery, an individual neuron here, with all of these different excitatory and inhibitory connections all around it distributed. It has to sum, or what they call summate, all of these different small voltage changes, these IPSPs and EPSPs, basically from around the cell, and make a decision, and see if it reaches the threshold at the hillock in order to initiate an action potential. So neurons actually summate these local EPSPs and IPSPs in two very critical ways. One is sort of spatial summation. So just to give you a sense, it's more these networks that you have developed through life are actually remarkably specialized and precise, in the sense that if there is an excitatory input here generating an EPSP, and one over here and one over here, so they're widely spatially distributed, well, they're unlikely to summate to minus 55 at the hillock, because they're going to be generating little IPSPs that are distributed pretty widely in space. However, if you've got lots of excitatory inputs that are coming in right here, this one location, you're more likely to generate significant depolarization here. The summation of multiple kind of contiguous, spatially close EPSPs, and that's more likely to generate an action potential. So that's spatial summation. The other, component obviously is time. Like if you have, 1,000 EPSPs, even if they're in the same location, but they're spread over a 24-hour period, you're unlikely to reach, minus 55 at the hillock and generate an action potential that'll propagate. However, if you've got lots of excitatory inputs coming in, very rapidly, temporally, very close to each other in terms of time, one right after the other, or at the same time, you're going to be more likely to summate to depolarization to the threshold that'll initiate an action potential down the length of the cell. So really, spatial and temporal aspects of activity are critical in determining whether a neuron will actually send an action potential or not.